What is the ladder of nature?
Aristotle, 384 to 322 b. CE, tried to use logic to build a diagram of living things. But he realized that living things could not be so easily apportioned. Instead, he constructed a ladder of nature or scale of perfection, which began with humans at one end and proceeded through animals and plants to minerals at the other end. Aristotle ranked these groups on a scale based on the four classical elements: fire, water, earth, and air.
In the life cycles of seedless, vascular plants such as ferns, is the sporophyte or gametophyte dominant?
The seedless, vascular plants exhibit an alternation of generations consisting of heteromorphic diploid and haploid phases, as found in all plants. The life cycle of seedless, vascular plants is dominated by the sporophyte. The sporophyte is the plant, for example, trees and flowers that everyone visualizes when they think of plants.
What is the function of bark?
Bark protects the interior tissues of a tree from external physical threats such as rain, hail, and snow, and biological threats such as animals, fungi, and bacteria. Bark also serves as a dumping ground in which the tree rids itself of waste products from its metabolism. Nutrients are transported through the phloem of the bark from the roots to the crown of the tree.
What is an enzyme?
Enzymes are proteins that act as biological catalysts. They decrease the amount of energy needed, activation energy, to start a metabolic reaction. Without enzymes, you would not be able to harvest energy and nutrients from your food. As an example, lactose intolerance is an inability to produce lactase, the enzyme that breaks down milk sugar, lactose. While this condition is not life-threatening for adults, it can have severe consequences for infants and children.
How are anabolic steroids harmful to those who use them?
Anabolic, protein building. Steroids are drugs that mimic the effects of testosterone and other male sex hormones. They can build muscle tissue, strengthen bone, and speed muscle recovery following exercise or injury. They are sometimes prescribed to treat some types of anemia as well as osteoporosis in postmenopausal women. Anabolic steroids have become a lightning rod of controversy in competitive sports. The drugs are banned from most organized competitions because of the dangers. They pose to health and to prevent athletes from gaining an unfair advantage. Adverse effects of anabolic steroids include hypertension, acne, edema, and damage to liver, heart, and adrenal glands. Psychiatric symptoms can include hallucinations, paranoid delusions, and manic episodes. In men, anabolic steroids can cause infertility, impotence, and premature balding. Women can develop masculine characteristics such as excessive hair growth. Male pattern balding, disruption of menstruation, and deepening of the voice. Children and adolescents can develop problems in growing bones, leading to short stature.
What antidote is available for mushroom poisoning?
No effective antidote for mushroom poisoning in humans has been discovered. The 138 toxins produced by mushrooms accumulate in the liver and lead to irreversible liver damage. Unfortunately, there may be no indication of poisoning for several hours after ingesting a toxic mushroom. When the symptoms do present, they often resemble typical food poisoning. Liver failure becomes apparent 3 to 6 days after ingesting a poisonous mushroom. Oftentimes a liver transplant may be the only possible form of treatment.
How is cell cloning used in scientific research?
Cell cloning is the process by which an exact copy is made of a cell. This cellular process is known as mitosis and is required for the growth and repair of multicellular organisms. Different types of cells in the body differ in their ability to perform mitosis. Some cells, like skin cells, produce clones quite often. Others, like those of the nervous system, will not reproduce after they have reached maturity and have differentiated. The scientific purpose of cloning is to produce many copies of certain types of cells that can then be used for a variety of purposes, like basic research or the growth of replacement organs.
What is nanotechnology?
The term nanotechnology was coined in 1974 by Norio Taniguchi, 1912 to 1999, at the University of Tokyo. It includes a number of technologies that deal with the miniaturization of existing technology down to the scale of a nanometer. 1/1000th of a meter, in size, about the size of molecules and atoms. Potential effects of nanotechnology include microcomputers capable of storing trillion of bytes of information in the size of a sugar cube, portable fluids containing nanobots that are programmed to destroy cancer cells, and airborne nanobots that are programmed to rebuild the thinning ozone layer.
What is kudzu?
Kudzu, pure Areolobata. is a vine that was brought from Japan for the 1876 Centennial Exposition in Philadelphia. It was intentionally planted throughout the southern United States during the 1930s in an attempt to control erosion. In fact, the federal government paid farmers as much as $8 an acre to plant it. In 1997, however, the government reversed its position on kudzu and referred to it as a noxious weed. Kudzu grows over everything that it encounters, draping itself across power poles and pine trees like a shawl. The plant is responsible for more than $50 million in lost farm and timber production each year. It grows at a rate of 120,000 acres per year. As of the early 21st century, it covers between 2 and 4 million acres of land throughout the United States, occurring from Connecticut in the east to Missouri and Oklahoma in the west, and south to Florida and Texas. Kudzu grows as fast as 1 foot 30 centimeters per day. The latest approach to controlling the growth of kudzu is to have goats chew on it, devouring the leaves, stems, and roots.
When were chromosomes first observed?
Chromosomes were observed as early as 1872, when Edmund Russo, 1841 to 1897, described seeing items that resembled small rods during cell division. He named the rod stab chen. Edouard van Beneden, 1846 to 1910, used the term batanet in 1875-40 to describe nuclear duplication. The following year, 1876, Edouard Balbiani, 1825 to 1899, described that at the time of cell division the nucleus dissolved into a collection of batanet citroids, narrow little rods. Walther Fleming, 1843 to 1905, discovered that the chromosomal threads or phadon split longitudinally during mitosis.
Can animals regenerate parts of their bodies?
Regeneration occurs in a wide variety of animals, however. It progressively declines as the animal species becomes more complex. Regeneration frequently occurs among primitive invertebrates. For example, a planarian, flatworm, can split symmetrically, with the two sides turning into clones of one another. In higher invertebrates, regeneration occurs in echinoderms, such as starfish, and arthropods such as insects and crustaceans. Starfish are known for their ability to develop into complete individuals from one cut-off arm. Regeneration of appendages, limbs, wings, and antennae occurs in insects such as cockroaches, fruit flies, and locusts, and in crustaceans such as lobsters, crabs, and crayfish. For example, regeneration of a crayfish's missing claw occurs at its next molt. Shedding of its hard cuticle exterior shell/skin in order to grow, and the subsequent hardening of a new cuticle exterior. Sometimes the regenerated claw does not achieve the same size of the missing claw. However, after every molt, a process that occurs 2 to 3 times a year, the regenerated claw grows and will eventually become nearly as large as the original claw. On a very limited basis, some amphibians and reptiles can replace a lost leg or tail.
What is the evolutionary time scale?
Trends in the fossil record require at least 1 million years to resolve themselves. And so paleontologists tend to work on a scale of 10 to 20 million years. Evolutionary biologists working on living species tend to describe selection as it occurs over a decade or less.
Is there an average lifespan of a cell?
No. Most living cells do not live longer than a month. Even cells that live longer, such as liver and brain cells, constantly renew their components so that no part of the cell is really more than a month old. One example of a cell with a longer lifespan is the memory B cell. Memory B cells can survive for several decades, even a lifetime, providing an immune response to pathogens invading for a second time.
Do male mosquitoes bite humans?
No. Male mosquitoes live on plant juices, sugary saps, and liquids arising from decomposition. They do not have a biting mouth that can penetrate human skin as female mosquitoes do. In some species, the females, who lay as many as 200 eggs, need blood to lay their eggs. These are the species that bite humans and other animals.
Can animals commit suicide?
There is no evidence of animals committing suicide. In strict Darwinian terms, this would be of no advantage to an individual's fitness.
What are sex chromosomes?
Sex, X and Y in mammals, chromosomes are the non-matching pair. The Y chromosome, found only in males, has the sex-determining gene, SRY. The SRY determines whether or not an individual will develop testes and produce appreciable quantities of testosterone, the hormone that generates male characteristics. There is no known correlate gene on the X chromosome for the formation of ovaries. Therefore, it is the absence of the Y chromosome that determines a female.
Besides humans, which vertebrates are the most intelligent?
According to the behavioral biologist Edward O. Wilson, 1929, the 10 most intelligent animals are the following: 1. Chimpanzee. 2 species, 2. Gorilla, 3. Orangutan, 4. Baboon, 7 species including drill and mandrill. 5. Gibbon, 7 species, 6. Monkey, many species, especially macaques, the patas. And the celeb's black ape, 7. Smaller toothed whale, several species, especially killer whale. 8. Dolphin, many of the approximately 80 species, 9. Elephant, 2 species, 10. Pig.
How are the results of paper chromatography deciphered?
Substances that have color are easily seen on a chromatogram. A completed chromatogram is sprayed with ninhydrin and may be heated to detect amino acids. Amino acids turn pink or purple when sprayed with ninhydrin. Molecules of reducing sugars become gray-black when treated with aniline phthalate. Many organic substances become brown spots on a yellow background when exposed to iodine vapors. The distance of the various spots from the origin is measured and used to quantify the movement of the sample. Under standardized conditions, a substance moves at a characteristic rate in a particular solvent system. A researcher can calculate the distance the solvent travels with the distance the substance travels. The result is known as the RF value. Consulting tables of RF values for various substances in various solvents permits preliminary identification of the substance.
Who proposed that Earth was only 6,000 years old?
In the 17th century, Archbishop James Usher, 1581–1656, of the Irish Protestant Church, calculated from the Old Testament genealogies that Earth was created on Sunday, October 26, 4004 CE. He was also able to calculate exact dates for when Adam and Eve were forced from paradise, Monday, November 10, 4004 BCE, and when Noah's Ark touched down on Mt Ararat, Wednesday, May 5, 1491 BCE.
What is the toxic release inventory?
Toxic Release Inventory, TRI, is a government-mandated, publicly available compilation of information on the release of over 650 individual toxic chemicals and toxic chemical categories by manufacturing facilities in the United States. The law requires manufacturers to state the amounts of chemicals they release directly to air, land, or water, or state that they transfer to off-site facilities that treat or dispose of wastes. The U.S. Environmental Protection Agency compiles these reports into an annual inventory and makes the information available in a computerized database. In 2000, 23,484 facilities released £7.1 billion, 3.2 billion kilograms, of toxic chemicals into the environment. Over 260 million pounds, 118 million kilograms of this total were released into surface water, 1.9 billion pounds. 86 million kilograms, were emitted into the air, over 4.13 billion pounds, 1.87 billion kilograms were released to land. And over 278 million pounds, 126 million kilograms were injected into underground wells. The total amount of toxic chemicals released in 2000 was 6.7% lower than the amount released in 1999.
What is a double-blind study?
In a double-blind study, neither the subjects of the experiment nor the persons administering the experiment know the critical aspects of the experiment. This method is used to guard against both experimenter bias and placebo effects.
How do fleas jump so far?
The jumping power of fleas comes both from strong leg muscles and from pads of a rubber-like protein called resilin. The resilin is located above the flea's hind legs. To jump, the flea crouches, squeezing the resilin, and then it relaxes certain muscles. Stored energy from the resilin works like a spring, launching the flea. A flea can jump well both vertically and horizontally. Some species can jump 150 times their own length. To match that record, a human would have to spring over the length of 2.25 football fields or the height of a 100-story building in a single bound. The common flea, Pulex irritans, has been known to jump 13.33 centimeters, in length and 7.25 centimeters, in height.
Why do we die without oxygen?
Most living organisms are aerobic, that is, they require oxygen to complete the total breakdown of glucose. As many as 36 ATP are produced through aerobic metabolism of one glucose molecule. Without oxygen, cells do not synthesize enough ATP to maintain a multicellular organism. Most people think that we need oxygen to breathe, but actually we need oxygen to recycle. The spent electrons and hydrogen ions, H+, produced as byproducts of aerobic respiration. With these byproducts, oxygen combines and forms metabolic water.
What is the ecological role of lichens?
Lichens account for approximately 8% of the vegetation covering Earth's surface. In certain environments, such as regions of tundra, they cover vast areas of land. Lichens delay global warming by consuming significant amounts of carbon dioxide. CO2, during photosynthesis. When they cover the ground, they prevent soil from drying out. In desert areas they are able to capture and conserve the moisture present in fog and dew. Lichens release nutrients, such as nitrogen and phosphorus, which are important in regions with nutrient-poor soils as the nutrients aid tree growth. Lichens are also an important food source for many species of animals, including wild turkeys and reindeer of the Arctic tundra. Birds such as the olive-headed weaver of Madagascar and the goldfinch of Europe use lichens to build their nests.
How is a fairy ring formed?
Long ago it was believed that the circles of mushrooms that sometimes form in meadows marked the locations where fairies gathered at night to dance. Fairy rings, or fungus rings, are frequently found in grassy areas. There are three types of rings: those that do not affect their surrounding vegetation, those that cause increased vegetational growth, and those that damage their surrounding environment. The rings are started from a mycelium, the underground, food-absorbing part of a fungus. The fungus growths are circular because a round inner band of decaying mycelium forms underground. This band uses up the resources present in the soil that is directly above it. When the fungus forms caps that present above ground, the mushrooms grow around the mycelium, creating a ring effect. Each succeeding generation grows further from the center.
What happens to the proteins present in an egg when it is cooked?
The white of an egg is rich in the protein albumin. When subjected to high heat, the bonds that form the three-dimensional structure of albumin are irreversibly changed. This causes the clear, jelly-like consistency of the egg to become firm and white. This process is known as protein denaturation. Proteins may also be denatured by a change in the pH value of the fluid surrounding them or by the addition of chemical detergents. Most denaturation is irreversible, however. Scientists can use partial or temporary denaturation to control the action of enzymes used in experiments.
What was the historical significance of hemp?
During the early years of colonial America, hemp, Agave cisalina, was as common as cotton is now. It was an easy crop to grow, requiring little water, no fertilizers, and no pesticides. The fabric looks and feels like linen. It was used for uniforms of soldiers, paper. The first two drafts of the Declaration of Independence were written on hemp paper, and an all-purpose fabric. Betsy Ross's flag was made of red, white, and blue hemp.
What is fish?
Fish, fluorescent in situ hybridization, is a method in which a clone gene is painted with a fluorescent dye and mapped to a chromosome. In fish, cells are arrested in the metaphase stage of mitosis and placed on a slide where they burst open, spreading chromosomes over the surface. A fluorescent labeled DNA probe of specific interest is placed on the slide and incubated long enough for hybridization to occur. The slide is then viewed under a fluorescence microscope that focuses ultraviolet light on the chromosomes. Hybridized regions of the chromosome fluoresce. This is a type of karyotypic analysis that can generate a physical map matching clones and gene markers to specific parts of a chromosome.
What are the dangers involved with transgenic animal transplants?
One of the major risks of xenotransplantation is the risk of transplanting animal viruses along with the transfer cells or organs. Since the patient is already immunosuppressed, the patient could die from the viral infection, or the virus could be spread to the general population.
How many antimicrobial products are available?
More than 8,000 antimicrobial products are registered with the U.S. Environmental Protection Agency, EPA. The EPA regulates products that kill microbes on inanimate surfaces. The various products contain more than 300 different active ingredients and are available as sprays, liquids, concentrated powders, and gases. More than 50% of the 8,000 antimicrobial products are used to control infectious microorganisms in healthcare settings.
What is the function of each of the different tissue systems?
The vascular tissue system consists of two kinds of conducting tissue, the xylem and the phloem. The xylem conducts water and dissolved minerals. The phloem conducts carbohydrates, mainly sucrose, hormones, amino acids, and other substances for the plant's growth and nutrition. The ground tissue system consists of three cell types: parenchyma, collenchyma, and sclerenchyma that have relatively thin walls and living protoplasts for storage, photosynthesis, and secretion. The dermal tissue system is the outer protective covering of the plant, including the cuticle.
What prevents urine from leaking out of the bladder and getting into the body?
The cells that form your bladder are held together by tight junctions, which are connections between cells that hold them together so closely that urine can't slip through to reach the rest of the body. These connections, formed by protein strands that bind the cell membranes, also play an important role in keeping food in the digestive tract until it has been completely processed.
How are coral reefs formed and how fast are they built?
Coral reefs grow only in warm, shallow water. The calcium carbonate skeletons of dead corals serve as a framework, upon which layers of successively younger animals attach themselves. Such accumulations, combined with rising water levels, slowly lead to the formation of reefs that can be hundreds of meters deep and long. The coral animal, or polyp, has a columnar form. Its lower end is attached to the hard floor of the reef, while the upper end is free to extend into the water. A whole colony consists of thousands of individuals. There are two kinds of corals, hard and soft, depending on the type of skeleton secreted. The polyps of hard corals deposit around themselves a solid skeleton of calcium carbonate, chalk, so most swimmers see only the skeleton of the coral. The animal is in a cup-like formation into which it withdraws during the daytime. The major reef builder in Florida and Caribbean waters, Montas triannularis. Star coral, requires about 100 years to form a reef just 3 feet 1 meter high.
What are the major groups of echinoderms?
There are 6 principal groups of echinoderms, from the Greek terms echina, meaning spiny, and derma, meaning skin. 1, class Crinoidea, sea lilies and feather stars. 2, class Asteroidea. Sea stars. 3, class Ophiuroidea, basket stars and brittle stars. 4, class Echinoidea. Sea urchins and sand dollars. 5, class Holothuroidea, sea cucumbers. And 6, class Concentricycloidea. Sea daisies, which live on waterlogged wood in the deep sea and were first discovered in 1986.
When were cells first studied?
The scientific study of cells developed gradually from the first description of cells in the 17th century. During this early period, investigations of cells were done using the primitive light microscopes available at the time and consisted almost entirely of morphological descriptions of cell structure. In the 18th and 19th centuries, research expanded to include the study of cell chemistry and physiology. Efforts that proceeded independently from morphological studies. The study of cell structure, cell chemistry, and cell physiology continued as separate fields of experimentation until the beginning of the 20th century, when the rapidly developing field of biochemistry began to influence cell biology. At the same time, genetics became established as a field of investigation.
Can bark be used to identify trees?
Tree bark varies in texture and color between species. It is used as an aid in identifying a particular species, especially when there are no leaves on the tree. Barks may be classified as to whether they are firmly attached to the tree trunk or whether they peel away from the surface of the trunk easily. Barks that are firmly attached to the tree trunk may have vertical fissures or furrows that may be deep or shallow. In other varieties of tree, the bark may look as if it is divided into squares or rectangles or other irregular shapes resembling scales. An example of scale bark is found in various species of the pine, genus Pinus. Shag bark has long overlapping thin sheets, such as is found in eucalyptus, genus Eucalyptus. Paper birch, Badala papyrifera, is an example of a species with bark that peels away from the tree easily, also known as ring bark.
What is xenotransplantation?
Xenotransplantation is the transplantation of tissue or organs from one species to another. The development of this technique has led to the breeding of animals specifically for use as human organ donors. Because humans would reject a non-human organ as foreign, transgenic animals, e.g., pigs, are genetically altered with human DNA with the hope of suppressing eventual rejection.
What are X-rays?
X-rays are electromagnetic radiation with short wavelengths, 10 to 3, and a great amount of energy. They were discovered in 1898 by William Conrad Röntgen, 1845 to 1923. X-rays are frequently used in medicine because they are able to pass through opaque, dense structures such as bone and form an image on a photographic plate.
How has centrifugation been used in biological applications?
Biologists primarily use centrifugation to isolate and determine the biological properties and functions of subcellular organelles and large molecules. They study the effects of centrifugal forces on cells, developing embryos, and protozoa. These techniques have allowed scientists to determine certain properties about cells, including surface tension, relative viscosity of the cytoplasm, and the spatial and functional interrelationship of cell organelles when redistributed in intact cells.
What is antisense technology?
Antisense technology involves targeting a protein at the RNA level as a means of regulating gene expression. The technology involves segments of DNA that are complementary to specific RNA targets. When the modified DNA binds to the RNA can no longer produce a protein. Antisense technology is a new technique in cancer therapy. Human trials are ongoing on small cell lung cancer and leukemia. Since cancer is a disease that can be characterized by excessive production of a particular protein, antisense technology works by interfering with the RNA that produces the protein.
In what ways are gymnosperms economically important?
Gymnosperms account for approximately 75% of the world's timber and a large amount of the wood pulp used to make paper. In North America, the white spruce, Picea glauca, is the main source of pulpwood used for newsprint and other paper. Other spruce wood is used to manufacture violins and similar string instruments because the wood produces a desired resonance. The Douglas fir, Pseudotsuga menziesii, provides more timber than any other North American tree species and produces some of the most desirable lumber in the world. The wood is strong and relatively free of knots. Uses for the wood include house framing, plywood production, structural beams, pulpwood, railroad ties, boxes, and crates. Since most naturally occurring areas of growth have been harvested, the Douglas fir is being grown in managed forests. The wood from the redwood Sequoia sempervirens is used for furniture, fences, posts, some construction, and has various garden uses. In addition to the wood and paper industry, gymnosperms are important in making resin and turpentine. Resin, the sticky substance in the resin canals of conifers, is a combination of turpentine, a solvent, and a waxy substance called rosin. Turpentine is an excellent paint and varnish solvent but is also used to make deodorants, shaving lotions, medications, and limonene lemon flavoring used in the food industry. Resin has many uses. It is used by baseball pitchers to improve their grip on the ball and by batters to improve their grip on the bat. Violinists apply resin to their bows to increase friction with the strings. Dancers apply resin to their shoes to improve their grip on the stage.
What effect do pH levels have on the growth of bacteria?
pH is the measure of the hydrogen ion activity of a solution. The pH scale ranges from 0, very acidic, to 14, extremely alkaline or basic. The pH, or concentration of hydrogen ions, H+, in an environment, is critical to bacterial growth because it can affect enzyme activity. An extremely high or low pH can denature and inactivate enzymes, or disrupt cell processes. An environment's pH level dramatically affects the growth of bacteria and other microorganisms. Each species has an optimum pH level to sustain growth, as well as a range of pH levels in which they are able to survive. Acidophiles have their growth optimum between pH 0 and 5.5, neutrophils, between pH 5.5 and 8.0. And alkalophiles prefer the pH range of 8.5 to 11.5. Extreme alkalophiles have optimum growth at pH 10 or higher. The following table shows the pH ranges and the optimum pH that several different organisms require for growth.
Why are people from certain geographic areas more likely to have different genetic traits?
Human activities can influence the distribution of alleles. Some examples include social constraints that limit choice of a mate to language, religion, and economic status, the tendency of some isolated populations, such as native island peoples, to stay isolated, and worldwide movement of populations due to technological advances. Studies have been done on the distribution of the B allele of the ABO red blood cell system. Initially, the highest occurrence of the B allele was in the indigenous populations of Central Asia. However, as people migrated from Asia into Europe, the frequency of the B allele changed, such that the highest frequency is now in Eastern Europe and lowest in Southwestern Europe.
How do neurons carry messages?
Imagine a long trail of dominoes running from your finger to the spinal cord in your back. When you touch a hot object, the messages this action produces in neurons is similar to the reaction that would happen if you knocked over the first domino of the set. After the first domino falls, each domino that follows it would be knocked over in turn. This process is similar to what happens between neurons. The message is carried from the fingertip all the way to the spinal cord. Neurons maintain a concentration of sodium and potassium ions, Na and Nk+, respectively. The openings present in membranes allow these ions to pass through. As the sodium and potassium gates open and then close, the electrical charge of the nerve cell changes and messages are transmitted along the neuron membrane.
Why was the Darwin-Wallace theory so ridiculed?
The work of Darwin and Wallace generated controversy on at least two fronts. First, their theory directly countered Christian teaching on the immutability of species and their special creation by God. Second, by presenting arguments for common descent. Darwin and Wallace showed that humans were related to other animals, apes in particular. This insulted those who felt that humans were unique and not part of the animal kingdom. However, it should be noted that as the work was disseminated among scientists, it gained general acceptance in the late decades of the 19th century.
What is cultural evolution?
Instead of changing at the molecular level, in the sequence of our genes. The history of Homo sapiens has been marked by what is rare in other animals, cultural evolution. Culture is passed from one generation to the next, and by changing our culture, we have been able to adapt to our environment without requiring structural changes. One example of cultural evolution is the emphasis placed on the use of soap and water to prevent disease. Adopting this cultural tradition has had a definite positive impact on the survival of human populations. And yet there have been no changes that would be visible in the fossil record of our species.
Who coined the phrase survival of the fittest?
Although frequently associated with Darwinism, this phrase was coined by Herbert Spencer. 1820 to 1903, an English sociologist. It is the process by which organisms that are less well adapted to their environment tend to perish and better adapted organisms tend to survive.
How are carbohydrates classified?
Carbohydrates are classified in several ways. Monosaccharides, single-unit sugars, are grouped by the number of carbon molecules they contain. Triose has 3, pentose has 5, and hexose has 6. Carbohydrates are also classified by their overall length. Monosaccharide, disaccharide, polysaccharide, or function. Examples of functional definitions are storage polysaccharides, glycogen and starch, which store energy, and structural polysaccharides. Cellulose and chitin, which provide support for organisms without a bony skeleton.
What are the two most common media to grow bacteria?
Beef extract and peptones, hydrolyzed protein, are the basic ingredients of nutrient broth. These materials supply a variety of carbon sources. Nitrogen compounds in the form of amino acids, and a mixture of cofactors such as vitamins. The addition of agar, a complex carbohydrate extracted from seaweed, results in a solid medium. Agar is an ideal solidifying agent for microbiological media because of its melting properties and because it has no nutritive value for the vast majority of bacteria. Solid agar melts at 194 to 212 degrees Fahrenheit, 90 to 100 degrees Celsius. Liquid agar solidifies at about 103 degrees Fahrenheit, 42 degrees Celsius.
What naturally occurring substance provides protection against viral infections?
Interferons protect the adjacent cells against viral penetration. Interferons are glycoproteins produced by body cells upon exposure to a virus. In 1957, Alec Isaacs, 1921 to 1967, and Jean Lindenmann, 1924, identified a group of over 20 substances that were later designated as alpha, beta, and gamma interferons.
Who is considered the first histologist?
Marcello Malpighi, 1628 to 1694, is considered the first histologist. For his pioneering work in the science of microscopic anatomy, he used the newly developed tool of the time, the microscope, to examine living things including both plants and animals. His observations included that blood passed through the capillaries and extensive work on insect larvae.
Do fungi exhibit an alternation of generations similar to plants?
Organisms with life cycles that exhibit an alternation of generations have both a haploid phase and a diploid phase. During the haploid phase, reproduction is conducted through gametes, which fuse together to form a zygote. During the diploid phase, reproduction is conducted by spores. Spores develop individually and divide through mitosis, producing the gametophytes of the next generation. Because fungi have both a haploid and diploid phase, they exhibit an alternation of generations.
Why would the honeybee genome be important?
The honeybee, Apis mellifera, is a major pollinator of crops and is a social insect, making it significant for basic research on insects.
How can you tell male and female lobsters apart?
The differences between male and female lobsters can only be seen when they are turned on their backs. In the male lobster, the two swimmerets, forked appendages used for swimming, nearest the carapace. The solid shell, are hard, sharp, and bony. In the female, the same swimmerets are soft and feathery. The female also has a receptacle that appears as a shield wedged between the third pair of walking legs. During mating, the male deposits sperm into this receptacle. where it remains for as long as several months until the female uses it to fertilize her eggs as they are laid.
What are the macronutrients and micronutrients of plants?
The macronutrients of plants are carbon, hydrogen, oxygen, nitrogen, potassium, calcium, phosphorus, magnesium, and sulfur. These all are nearly or in some cases far greater than 1% of the dry weight of a plant. The micronutrients are iron, chlorine, copper, manganese, zinc, molybdenum, and boron. Each of the micronutrients constitutes less than 1 to several hundred parts per million in plants. Sodium, silicon, cobalt, and selenium are beneficial elements. Research has not shown that these elements are essential for plant growth and development.
How are animals classified?
Animals belong to the kingdom Animalia. Most biologists divide the kingdom into two subkingdoms. 1. Parazoa. From the Greek para, meaning alongside, and zoa, meaning animal, and 2. Eumetazoa. From the Greek eu, meaning true, meta, meaning later, and zoa, meaning animal. The only existing animals classified as parazoa are the sponges, phylum Porifera. Sponges are very different from other animals and function much like colonial unicellular protozoa even though they are multicellular. Cells of sponges can be versatile and change form and function and are not organized into tissues and organs. They also lack symmetry. All other animals have true tissues, are symmetrical, and are classified as eumetazoa.
Are freshwater clams an endangered group?
Although freshwater clams are found on every continent except Antarctica, they are now considered one of the most jeopardized groups of animals in the world. Approximately 270 species belong to the family Unionidae, found in North America. A total of 72% of our 270 native mussel species are listed as recently extinct. Endangered, threatened, or of special concern due to human impact on aquatic habitat, commercial harvesting. The introduction of carp, water pollution, and the invasion of zebra mussels.
Who was Comte de Buffon?
Georges Louis Leclerc, Comte de Buffon, 1707 to 1788, was an early proponent of natural history, which is the study of plants and animals in their natural settings. He was also known for his work as a mathematician. Buffon was interested in the modes by which evolutionary change could occur. A prolific writer, his work Natural History comprised 35 volumes. He pondered the meaning of the term species and whether such groupings were immutable, unchanging, over time. In addition, he served as mentor to Jean-Baptiste de Lamarck, 1744 to 1829.
What is lyophilization?
Lyophilization is a freeze-drying technique for preservation and storage of bacteria and other microorganisms. Bacteria can be stored for extended periods of time, 3 to 5 years, as frozen cell suspensions or as freeze-dried, lyophilized, cultures. This technique is achieved by placing bacteria in a nutrient broth containing 15 to 25% glycerol and freezing at temperatures of -94 degrees Fahrenheit -70 degrees Celsius or lower. The glycerol reduces ice crystal formation that would cause subsequent cell damage and disrupt biological structures.
Are all cell membranes alike?
Although all cell membranes have the same general structure, the membrane composition is different for each species, depending on their function. Membranes vary in the amount of protein they contain or the type of membrane receptors they contain. For example, plants that can survive frigid temperatures, such as winter wheat, are able to increase their concentration of unsaturated phospholipids during the winter to prevent membrane solidification. Another example is muscle cells, which have plasma membrane receptors for the neurotransmitter acetylcholine that tells them when to contract.
What happens to proteins during a hair perm?
During a hair perm, the bonds that form the secondary and tertiary levels of structure in the protein keratin, the major protein of hair, are chemically broken and then reformed. Hydrogen bonds are primarily involved in this process. The same principle, breaking and forming bonds, is used to make hair either very curly or very straight.
What are the four major groups of plants?
Plants are divided into phyla based on whether they are vascular, containing vascular tissue consisting of cells joined into tubes that transport water and nutrients, or non-vascular. The phyla of vascular plants are then further divided into seedless plants and those that contain seeds. Plants with seeds are divided into flowering and non-flowering groups. Non-vascular plants have traditionally been called bryophytes. Because bryophytes lack a system for conducting water and nutrients, they are restricted in size and live in moist areas close to the ground. Examples of bryophytes are mosses, liverworts, and hornworts. Examples of seedless, vascular plants are ferns, horsetails, and club mosses. The conifers, which are cone-bearing, are seed-bearing, non-flowering vascular plants. The majority of plants are seed-bearing, flowering, vascular plants known as angiosperms.
How many classes of internal membrane-bound structures are found in eukaryotic cells?
There are two classes of internal membrane-bound structures in eukaryotic cells. There are discrete organelles such as mitochondria, chloroplasts, and peroxisomes. Then there is the dynamic endomembrane system: nuclear membrane, endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles.
What happens if a cell's DNA is mutated?
DNA contains the code for building everything within the cell. If the DNA code is changed, or mutated, the cell will build things differently. Mutations can change the way that a cell produces substances, such as proteins and carbohydrates, builds organelles, or responds to messages. These changes can enhance a cell's ability to survive. Although it is more likely that drastic changes will result in a decrease of efficiency or even the death of the cell.
What are Koch's postulates?
Koch's postulates are the four basic criteria an organism must meet in order to be identified as pathogenic, capable of causing disease. The characteristics are as follows: 1. The organism must be found in tissues of animals that have been infected with the disease, rather than in disease-free animals. 2. The organism must be isolated from the diseased animal and grown in a pure culture or in vitro. 3. The cultured organism must be able to be transferred to a healthy animal, which will show signs of the disease after having been exposed to the organism. 4. The organism must be able to be isolated from the infected animal.
What is the pollutant standard index?
The U.S. Environmental Protection Agency and the South Coast Air Quality Management District of El Monte, California, devised the Pollutant Standard Index to monitor concentrations of pollutants in the air and inform the public concerning related health effects. The scale, which measures the amount of pollution in parts per million, has been in use nationwide since 1978.
Who was Linus Pauling?
Linus Carl Pauling, 1901 to 1994, the only person to win two unshared Nobel Prizes. 1954 Prize in Chemistry, 1962 Peace Prize, revolutionized the study of chemistry. Aided the development of molecular biology, and made important advances in medical research. He was responsible for determining the molecular basis of sickle cell anemia. Was one of the scientists engaged in the race to determine the molecular structure of DNA. And became a strident advocate for the use of large doses of vitamin C to prevent illness.
How many Golgi bodies are in a cell?
Protists contain one or a small number of Golgi bodies. Animal cells may contain 20 or more Golgi bodies, while plant cells may contain several hundred Golgi bodies.
What are examples of growth factors that affect cell division?
In order for a cell to grow, it must have specific nutrients. Some cells may require a messenger referred to as a growth factor to stimulate growth. There are several classes of proteins known as growth factors, which may also influence other cell activities, including embryonic development and response to tissue injury.
What is the only four-horned animal in the world?
The four-horned antelope, Tetracerus quadricornis, is a native of central India. The males have two short horns, usually 4 in, 10 centimeters, in length, between their ears, and an even shorter pair, 1 to 2 in, 2.5 to 5 centimeters, long, between the brow ridges over their eyes. Not all males have 4 horns, and in some the second pair eventually falls off. The females have no horns at all.
What are nectaries?
Plants secrete a variety of substances from specialized structures called secretory structures. Nectaries are structures that secrete nectar, a sugary compound that attracts insects, birds, or other animals. Most nectaries are associated with flowers and are called floral nectaries. Nectar is 10 to 50% sugar, especially sucrose, glucose, and fructose. Plants usually produce small amounts of nectar, which forces foraging animals to visit several flowers before obtaining a full meal. A single insect or bird can, therefore, pollinate tens or hundreds of plants.